That's my problem. I know this shouldn't be distracting. I was struggling to do some like reading, but I was like, okay. It was just the two videos. Easier as like a warm up or like there's like my max that I've done.
Good morning, everyone. How's everybody doing today? Good. Okay, great. So the topic for today is something that we've alluded to in a lot of the medical biochemistry sessions so far, the TCA cycle. So over there in the gray or beige section, the little wheel. So I think that's a very important central node in metabolism, so hopefully it's going to be pretty helpful to have that deeper understanding of that by the end of the 2 hours. So we have a few different pathologies for today's lectures. Understand these in a general sense. These mitochondrial conditions are pretty tricky to diagnose, and their pathologies can be really variable. But mainly, I'd like you to understand the rationale for why that presentation can be variable and like the general hallmarks of those conditions. Okay. Should we start with a little clicker quiz? These will be some pretty simple questions, but this is a review one. The rest, many of them will kind of lead into discussions that we'll have over the next 2 hours. Everybody had a chance to ponder this one? Okay, acyl-CoA dehydrogenase is the right answer. So CPT1, that's a good guess because that's the rate limiting step for fatty acid beta oxidation, but this is a step below that, or 2 steps below that. So this is a one-step question. Just, I don't have an example of this, but what could I do to this to make it into a 2-step question? What's one thought?
You could describe like the drinking vomiting illness and then ask what exactly.
Precisely, yep. Yep, we could give a clinical presentation and a diet history of eating the ackee fruit or something, and then ask what enzyme is inhibited. Yeah, good. Okay. Has Dr. Cormier talked about epigenetics a little bit? Okay. Okay. Should we take a look here? And this is a kind of a universal methyl donor, S-adenosylmethionine. So that's the proximal substrate from a lot of methyltransferase reactions. Now, S-adenosylmethionine is dependent on what the majority, a bare majority of you selected, vitamin B12, S-adenosylcobalamin. Okay, so cobalamin provides the methyl for S-adenosylmethionine, then methionine is the substrate for the methyltransferases. Okay, then how about something totally different? And this isn't a, of course, on primate phylogeny, but essentially you've got 4 primates and 1 non-primate there. Okay. Okay, 100%, awesome. Now, this is a true statement, however, there's an exception to this in every eukaryotic cell, and that is the mitochondrial genome. Okay, so just kind of keep that in the back of your mind. We'll discuss properties of mitochondrial DNA very shortly. Okay, shall we start with the didactic portion of the lecture? Okay. All right, so the topic of today is the TCA cycle. And the main function of the TCA cycle in the fuel oxidation sense is to take the 2 carbons of acetyl-CoA and turn those 2 carbons into carbon dioxide. And then we generate GTP and reducing equivalents in the form of NADH and FAD2H to power the electron transport chain. Okay, so this is in the wheel portion of our Stanford map of human metabolism. As a high altitude function of the TCA cycle, it participates in fuel oxidation through that conversion of acetyl-CoA to carbon dioxide. It also is a metabolic hub for many biosynthetic reactions, including amino acid synthesis, heme synthesis, nucleotide synthesis, what have you. It spits out a lot of different substrates for biosynthetic reactions. It can also take in a lot of different substrates aside from acetyl-CoA for anaplerotic reactions, as we'll discuss. Okay, so the tricarboxylic acid cycle, this is also called the citric acid cycle or the Krebs cycle. You may notice, hear it referred to that. I'm going to always try to call it the TCA cycle. Okay. All right, so the TCA cycle is a mitochondrial metabolic pathway. So I'm just going to take a few moments to discuss the general properties of the mitochondria. And these are important for both transport of substrates into and out of the mitochondria for the TCA cycle. It's also very important for the subject of Friday's lecture, the electron transport chain. So there'll be some overlap. Okay, so we can see a representation from a electron micrograph of a mitochondria. It's a little bit useful to, or more useful to look at the cartoon representation. But essentially, the mitochondria has a membrane structure that's not like any other membrane within the cell. So the outer mitochondrial membrane is similar in phospholipid composition to plasma membrane or nuclear envelope or ER or what have you. The inner mitochondrial membrane, this is very impermeable relative to other cellular membranes, and it's impermeable because it uses, rather than the typical phospholipids, it uses this really large phospholipid called cardiolipan that's like a kind of a small polymer of phospholipids, if you will. So the transport of things from the cytoplasm into this intermembrane space is relatively easy, but very few things can cross the inner mitochondrial membrane without a protein carrier. Okay. So the functional consequences of this is that the cell can set up concentration gradients for protons, for example, between the mitochondrial matrix, the interior side, and this intramembrane space. So concentration gradients of protons and concentration gradients of charge or charge differentials across that inner mitochondrial membrane that can be used to power ATP synthesis. Okay, and it also powers some transport processes for things in and out of the mitochondria. Okay, so that's kind of the general organization of the mitochondria, and the mitochondrial capacity of any given cell is going to be dependent on the cell's function and the cell's environment, and we'll have an example of that in a little bit. So mitochondria have their own DNA, and the DNA is quite different from nuclear DNA. It's circular, first of all, and the copy number of the circular DNA per mitochondria and per cell is variable. So unlike the nuclear DNA, where it's under normal circumstances, there are only 2 copies of each chromosome, right? And errors in cell division can cause exceptions to that, but under normal circumstances, you only have 2 chromosomes per 2 copies of each chromosome per cell. With the mitochondrial genome, that's different. You can have several dozens or several thousands of copies of the mitochondrial genome per cell. Furthermore, you can have multiple copies of the mitochondrial genome within each mitochondria. Okay, so what's encoded on the mitochondrial genome? The mitochondrial genome encodes some subunits of the electron transport chain proteins. So those are shown here: complex 2, 3, 4, and then ATP synthase is complex 5. So each of these are enzymes that are composed of multiple protein subunits, and some of those subunits are encoded on the mitochondrial genome. The exception is complex 2, succinate dehydrogenase. This is an enzyme that participates in electron transport chain and also in the TCA cycle. And the succinate dehydrogenase, this is the only enzyme in the electron transport chain that doesn't have any part of its protein encoded by mitochondrial genome. These are all nuclear genes. So shown here, the blue subunits in each one, these are representing nuclear encoded subunits, and then the different colored subunits are representing mitochondrial subunits. Okay, so in addition to those encoding the critical components of the electron transport chain, the mitochondrial genome also has everything else that's needed for expression of those genes encoded onto the mitochondrial genome. So if you think, like, what do you have to express genes, you're going to need ribosomes, and there are 2 ribosomal RNAs that are encoded on the mitochondrial genome, and then transfer RNAs. So all of the necessary transfer RNAs for each amino acid is also encoded on the mitochondrial genome. So the representing here, like, this is the origin of replication and the origin of transcription, and then each of the little colored things here represents a coding region for a protein subunit of the electron transport chain, and then like H, that would be the transfer RNA for histidine. Okay, so it's got each one of those encoded on it. Mitochondrial DNA also, in addition to just one more unique little bit of trivia about the mitochondrial DNA, it's inherited maternally. So there's maybe some exceptions to this, but I think it's safe to say that mitochondrial DNA is inherited exclusively from the mother. Okay, so the mitochondrial DNA encodes key subunits of the electron transport chain, and the expression of those genes is similar to how a bacteria might express its genome. So there's one transcript that's generated through the transcription through the transcriptional start site, and you can get this rolling circle type. Have you heard that for bacterial DNA replication? So you can get the mitochondrial RNA polymerase kind of spinning around and generating multiple polycistronic mRNAs that each encode encode the entire sequence of enzymes, plus the transfer RNAs, and those are processed and used as templates for the ribosomes. Okay, so one of the features of the mitochondrial genome that make it similar to bacterial genome is this polycistronic gene expression. Other features are the circularity of it, and the gene density is, if you look, I mean, there's like no non-coding DNA there, hardly any, right? It's either coding for a gene or a transfer RNA, or it's a origin of replication, okay? Whereas, as we know with the nuclear DNA, there are billions and billions of bases that, you know, they used to be considered junk DNA. I think that's maybe going away, especially if you, has Dr. Lotcher mentioned that? Okay, if don't get her started on it. But so, anyways, it's non-protein coding DNA, right? Let's just put it that way and not be judgmental about it. Okay, so furthermore to this comparison of mitochondrial DNA with nuclear DNA, mitochondrial DNA is hypermutable compared to nuclear DNA. And the reasons for that are, first of all, it's in a big environment of oxidative stress. So most of your oxidative stress in normal circumstances comes from superoxide that's generated from the mitochondria. So the mitochondrial DNA is right proximal to the biggest source of oxidative stress. Beyond that, the chromatin structure, if you will, of mitochondrial DNA is much less robust than the chromatin structure of nuclear DNA. I think I have that it's naked here. So mitochondrial DNA compared to nuclear DNA, it's got less clothes on, I guess, than nuclear DNA. It's maybe not appropriate to say that it's naked. There are mitochondrial DNA binding proteins, but they're much looser attached than that tight chromatin structure around the histones. Then the final thing that makes the mitochondrial DNA hypermutable is that the mitochondrial DNA polymerase gamma, that doesn't have the same proofreading capacity as the nuclear DNA. Okay, so we've got more errors that are accumulating, and due to more errors happening from reactive oxygen species and the lack of protection of the DNA, and then errors of replication can't be corrected as robustly as in the nuclear DNA. Now, this hypermutability is balanced by the variable copy number of mitochondrial genomes. Okay, if you get a mutation in a nuclear gene, then you're automatically heterozygous, right? At least half of your DNA content for that gene is mutant. If you've got one copy of the gene is mutant and the other is wild type. With mitochondrial DNA, if you have 100 copies of your mitochondrial DNA in a cell, you can have 1% of it be mutant and 99% of it be wild type. Or the converse is also true. Okay, you could have most of it be mutant and some of it be, and a small amount be wild type. So that mitochondrial DNA elevated copy number, that could act as a sink maybe to kind of counterbalance this hypermutability. Okay, but it has some consequences for the presentation of mitochondrial diseases or diseases that result from mutations in the mitochondrial DNA. So mitochondrial DNA copy number will vary from one cell type to another and one individual to another. The mitochondrial copy number within any cell type is also dynamic. So this is showing that the mitochondrial DNA copy number can be pharmacologically manipulated. In this case, there's two of the PPAR gamma agonist glitazone drugs. I think the pioglitazone, this is still on the market as a anti-diabetes drug or anti-metabolic syndrome drug. So these are different cells that are cultured in a somewhat normal glucose concentration and then a hyperglycemic concentration. And then the mitochondrial copy number per cell, or the mitochondrial copy number per nuclear gene copy number is shown on the y-axis. So just hyperglycemia doesn't affect the mitochondrial DNA copy number, but by activating the PPAR gamma transcription factors with these pharmacological agents, you can get a program on the cell, a transcriptional program that promotes more mitochondrial capacity, and this then gives you more oxidative metabolism and can be beneficial in the for hyperglycemia. Okay, so don't worry so much about that, but just the principle is that mitochondrial DNA copy number is not fixed and can be manipulated pharmacologically and through exercise and what have you. Okay, so mitochondrial diseases, this is referring to pathologies that are associated with mutations in the mitochondrial DNA. And these can have a quite variable presentation because of that different mode of inheritance of the mitochondrial DNA. So if you could imagine a situation, so you'll talk about oogenesis and early development, I think, from Dr. Holy later in the course, if you haven't already. So the details of this he'll elaborate on. But anyway, in early oogenesis, if you could imagine, this is a mother with a mitochondrial disease that's affecting 50% of the mitochondrial DNA in the early oocyte. Now, as that oocyte is dividing, the distribution of the mutant versus the wild type mitochondrial DNA is not going to be 50-50 in each of the daughter cells. Okay, just by chance, you may end up with a daughter cell that has a high dose of the mutant DNA or a very low dose of the mutant DNA. Okay, so this then is going to affect the, this is going to affect both the presentation of the condition, so it may be completely silent if there's a low gene density, and it's also going to affect the ability of the offspring, if it's a female, to pass that condition on to any children that she may have. So does that make sense? Okay. Okay, so I have here a table of some of the mitochondrial diseases that have been described for a long time. Don't commit this to memory, but just kind of appreciate that there are some common symptoms of progressivity. So these tend to be exacerbated or get worse rather than getting better, and that's because of your mitochondria are always getting worse, unfortunately, because of that hypermutability of the mitochondrial DNA. And then if you overlap a poorly functioning mitochondria on top of that, it's going to progress. And these tend to affect neuronal tissues as kind of one of their hallmark symptoms. So you'll get seizures or epilepsy or deafness or symptoms that will manifest from loss of function of neurons of some sort. Let's see. I'd just like to consider, so the myoclonic epilepsy and ragged red fiber condition, this is caused by a point mutation in the lysine transfer RNA, right? So please consider what would be the consequences on gene expression of mitochondrial proteins in the context of this point mutation. Which proteins would be affected? So anywhere, okay, so because the mitochondrial encoded subunits of the electron transport chain, they're all encoded in um, the mitochondrial DNA, right? Or the key ones are encoded in mitochondrial DNA. So anytime the message got to the lysine codon, the transfer RNA would be absent and the translation would stall. So this would be the case for the subunits of the electron transport chain that are encoded only on the mitochondrial genome. Right, there would still be normal proteins that are encoded by the nucleus and translated in by ribosomes and then imported into the mitochondria. Those could have all the lysine they want because they're depending on different transfer RNAs. Okay, now it gets even more confusing because, say, you have 50% mutant DNA in your mitochondria and 50% normal DNA. You're still going to get some translation of the mitochondrial DNA encoded genes, but it'll be compromised. Okay, so these mitochondrial diseases that are caused by the mutations in the mitochondrial genome are difficult to diagnose, and they have very variable presentation. Okay, all right. So let's move on to the TCA cycle. And we're going to, well, I'm going to have one little diversion back before we get to the TCA cycle. And you're going to get tired of me talking about pyruvate dehydrogenase, but unfortunately we got to talk about it, just one other topic on pyruvate dehydrogenase. Okay, so the TCA cycle functions in its fuel oxidation capacity. It functions to take acetyl-CoA and take the 2 carbons from the acetate and dump them off as carbon dioxide, and then we get energy in the form of GTP, a guanosine triphosphate that's energetically equivalent to an ATP, and then our reducing equivalents, which we can transfer to the electron transport chain to make more ATP. So these two things are when the cycle is going in the clockwise direction in the fuel oxidation capacity. We can also, in the anabolic capacity or biosynthesis capacity, we can generate precursors for a lot of other different things. Okay, so the substrate for the TCA cycle in its fuel oxidation capacity is acetyl-CoA. We've seen how we can get acetyl-CoA from diverse catabolic processes. One of the most important regulators of which of those substrates is going into the TCA cycle is the pyruvate dehydrogenase complex, and we've seen that, as a model of cofactor utilization and maybe in one other context. So the PDH, this is the enzymatic function is to take pyruvate and oxidatively decarboxylate it into acetyl-CoA, and this is really regulating which substrate in the cell is being used to power the TCA cycle. So when PDH is on, the cell is going to be preferentially using carbohydrate. When PDH is shut off, then the cell is going to be switching over into fatty acid catabolism, fatty acid beta oxidation, or amino acid catabolism. Okay, so the fatty acid catabolism, that's also regulated by CPT1 activity, but these are going to be regulated in a coordinate fashion. Okay, so the PDH, pyruvate dehydrogenase, that is present in the mitochondria like all the TCA cycle enzymes. So in order for the substrate pyruvate to get from where it's made in the cytoplasm through glycolysis into the mitochondria, it has to go through a dedicated pyruvate carrier called the mitochondrial pyruvate carrier, which is a 2-protein complex that was identified just about 10 years ago. So remember, the intermitochondrial membrane, the transport across it is highly regulated, so you've got to go through this protein carrier. This family these two genes that encode the mitochondrial pyruvate carrier, these were identified in a family that had inherited lactic acidosis. Okay, so we imagine how that might happen? So let's see, we've got glucose coming in here, being fermented to, or used, glycolysis converts it to pyruvate. Then it goes through the mitochondrial pyruvate carrier into the mitochondria. Okay, and then it's completely oxidized. If you knock this out, then one of the metabolic fates for the pyruvate then is to go through the lactate dehydrogenase reaction and be exported out through the monocarboxylate transporter. So in the case of a mutation of the mitochondrial pyruvate carrier, you've got kind of two things occurring. You've got energy starvation because you can't get the maximum amount of energy from carbohydrate metabolism. You could still be doing fatty acid beta oxidation, but this way to get substrate for the TCA cycle would be absent. Because of that energy starvation, you're going to increase glycolysis, and the pyruvate that's generated from glycolysis doesn't have a metabolic fate except to go to lactate. Okay, so that can then cause acidification of the extracellular space and cause muscle pain and ultimately death if it's not corrected. Okay, so now our pyruvate has gone from glycolysis into the cytoplasm and is in the mitochondria, so it's a substrate for the pyruvate dehydrogenase complex. Recall that pyruvate dehydrogenase is this multi-subunit enzyme, and each component of the machine has a different enzymatic function and a different cofactor requirement. Right, so just reviewing that, and that's actually going to come back when we talk about alpha-ketoglutarate dehydrogenase. Okay, so the regulation of the pyruvate dehydrogenase complex is at the level of reversible phosphorylation of serine amino acids on the E1 subunit. So this is the thymine pyrophosphate binding subunit that catalyzes the decarboxylation component of the reaction. So there are three, I've just shown here, a single phosphate, but there's actually three serine amino acids on the E1 subunit, and they are phosphorylated by a family of pyruvate dehydrogenase kinases, and these are activated in conditions of energy excess. So a lot of acetyl-CoA, a lot of reduced NADH, or a lot of ATP will cause them to be transcriptionally activated. Then kind of the converse, they're also activated in conditions of hypoxia, because if you don't have oxygen for the electron transport chain, you shouldn't be putting substrates and reducing equivalents into the TCA cycle. Okay, so there's kind of two conceptually different things that are activating the kinases. And when the kinases are phosphorylating the E1 subunit, that's inactivating the enzyme, preventing pyruvate from going into the TCA cycle. In that case, the cell has to try to switch to either anaerobic glycolysis or using fatty acid or amino acid oxidation for fuel. There are also a family of phosphatases, two phosphatases that will remove that inhibitory phosphate. These are activated by calcium, and calcium, that's signaling is associated with intracellular work. For example, muscle contraction will cause pulses of calcium that will be associated with work being done and need for energy, so you want to get substrates into the TCA cycle. Okay, so that's just showing a little bit of the regulation of this pathway, and when this pathway is regulated, that's going to impact the other pathways. Okay, so let's review or have an overview of the TCA cycle, and then I'll go through reaction by reaction, and then we'll talk about a few key steps there. Okay, so these are all mitochondrial reactions, and we can, let's look at this first in the fuel oxidation function. That is, we're taking 2 carbons from acetates and converting those into carbon dioxide. So just going from this way to this way. Now, we can function in reverse. Things can go out of it and into it, but for now, let's just consider it as a linear pathway from oxaloacetate back to oxaloacetate. Okay, so in that fuel oxidation function, the key regulatory step is isocitrate dehydrogenase. This is a enzyme is allosterically activated by ADP, activated by citrates, and we'll talk about the details of that. But essentially, the activity of isocitrate dehydrogenase will determine the activity of everything else, or the activity of the entire cycle. Now, when isocitrate dehydrogenase is, say, inactive within a cell, if this activity is blocked, we might logically predict that isocitrate would build up, right? Now, it does, but if your cycle is not moving forward, isocitrate builds up, but then it's converted back into citrate. And that's because of, if you think back onto, it must have been 3 or 4 months ago, we talked about delta Gs, right? Okay, so the delta Gs, the delta G of the entire cycle is driven by some large negative delta Gs on the citrate synthase and isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase step. So the green arrows here, these have large negative delta G0 primes. So these steps are, reversible under physiological conditions, and I'll show an example of the fumarate hydratase reaction, how that's reversible. That's kind of interesting. Okay, so the conversion of citrate to isocitrate, this has a positive delta G0 prime. And the consequence of this within cells is that if we have a block in the isocitrate dehydrogenase step, we don't get an accumulation of isocitrate, we get an accumulation of citrate because the reaction kind of energetically falls backwards towards citrate. Similarly, if the cycle is blocked here, we don't get a buildup of oxaloacetate, we get a buildup of malate. Okay, and this is useful because then the citrate can divert, or the malates can divert out to other pathways. Okay, so let's consider reactions one by one, and again, we're just starting from acetyl-CoA and oxaloacetate, and then we're going to end with oxaloacetate. Okay, so the first reaction is catalyzed by citrate synthase, and it's taking the 4-carbon oxaloacetate and joining that with the 2 carbons from acetyl-CoA to make the citrate molecule, gesundheit. Citrate is a tricarboxylic acid, so this is a carboxylic acid, this is a carboxylic acid, and this is a carboxylic acid. So that's where the name comes from. Okay, easy enough. The next step is a rearrangement of that alcohol group on citrate, and this is a 2-step dehydration and hydration reaction catalyzed by aconitase. So the alcohol starts there, goes off as water, then comes back from water, except it's on a different carbon to make isocitrate dehydrogenase. Okay, so isocitrate dehydrogenase then, that is the rate-limiting step in the TCA cycle. There are 3 different isocitrate dehydrogenase enzymes. The TCA cycle enzyme is referred to as IDH3. It's also referred to as NAD+ dependent isocitrate dehydrogenase. The other 2, IDH1 and IDH2, they're using NADP as an electron acceptor. The TCA cycle version is using NAD+. So this is a decarboxylation reaction. So isocitrate, this is the tricarboxylic acid. It's losing that branched carboxylic acid as carbon dioxide, and the product is the keto dicarboxylate alpha ketoglutarate. Okay. And when we're doing this, we're transferring electrons from this carbon to NAD plus to make NADH. So this is an energy generating step within the TCA cycle. And just a little bit more detail about how that reaction happens. And remember our niacin, this is the redox component or the redox location for our electron acceptor. Yeah, I think that's all that I wanted to say that. Now, the next enzyme, alpha ketoglutarate dehydrogenase, also participates in a decarboxylation reaction. Okay, but it's a different reaction, and well, okay, I don't want to get ahead of myself, but so the alpha ketoglutarate dehydrogenase, that's taking another carbon or a carboxylic acid group from our substrate and dumping that off as carbon dioxide. We have then a dicarboxylic acid, so a carboxylic acid there, and now we have a coenzyme A added in to bind the substrate. Okay, so this is alpha ketoglutarate dehydrogenase, and alpha ketoglutarate dehydrogenase has a familiar, hopefully familiar, subunit organization and cofactor requirement. So it's participating in a oxidative decarboxylation, removing carbon dioxide from a substrate, then transferring the rest of the substrate to coenzyme A. So it's using the same thymine pyrophosphate and lipoic acid and FAD and NAD+, same reaction as pyruvate dehydrogenase. Okay, now I'm going to go back a section here. Isocitrate dehydrogenase doesn't have that same subunit composition and doesn't use thymine pyrophosphate. So what's different about the isocitrate dehydrogenase reaction compared to the alpha ketoglutarate dehydrogenase reaction? Yeah.
The involvement of acetyl-CoA.
That's one thing. Yep, that's definitely one thing. So the pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase, they have an acyl transferase function that they're taking the carbon substrate and joining it to coenzyme A. Isocitrate dehydrogenase, the product is not a CoA ester, right? It's just alpha ketoglutarate out in solution. Then another, so yeah, that's one key difference. Another key difference is that the substrates for alpha ketoglutarate dehydrogenase is a keto acid. It's a keto dicarboxylate. The isocitrate dehydrogenase is an alcohol tricarboxylate. Okay, so that there are three enzymes I can think of: the pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, and then remember the branched chain keto acid dehydrogenases that are important for maple syrup urine disease. So they all perform that oxidative decarboxylation and making a CoA ester with the product, and they all have the same basic architecture and subunit composition and cofactor requirements as shown here. Okay, so kind of interesting. Okay, so now we have succinyl-CoA, and succinyl-CoA is a substrate for succinate thiokinase. This is an energy generating step within the TCA cycle where the high energy bond between succinate and the coenzyme A, This is broken, and the energy is used to catalyze the addition of inorganic phosphate to GDP to make guanosine triphosphate. Then the dicarboxylate succinates, this is a product. Succinate dehydrogenase then is removing electrons that are shared by these carbons in a single bond to make a double bond fumarate. And the electrons then are transferred to FAD to reduce it to FAD2H. The electrons then can be transferred directly to coenzyme Q, and coenzyme Q can transfer the electrons to complex 3 and complex 4, et cetera, to generate ATP in the electron transport chain. So succinate dehydrogenase, this is considered complex 2 in the electron transport chain, and it's also considered, the 6 o'clock point in the metabolic map, and so the 6 o'clock point in the metabolic map, that's making a tangent with the electron transport chain. So it's performing 2 functions. Okay, my computer's asking me for an update here at the best time. Okay, so fumarate then, fumarate is a substrate for fumarase, or this is also, I think your textbook calls this fumarate hydratase. So this is the conversion of this double bond into an alcohol group with the addition of water. This is actually, I've only got the arrows going one direction here, but this is a reversible reaction. And it's reversible within the mitochondria, and it's also reversible in the nucleus and has an important role in DNA repair. So this is kind of an out there link between the TCA cycle and some of Dr. Cormier's material on DNA repair. Okay. The final enzyme is malate dehydrogenase, and malate dehydrogenase is going to convert this alcohol into a ketone and regenerate oxaloacetate. And as it's doing this, it's reducing an NAD plus to NADH. Okay, so now we're back at oxaloacetate, and citrate synthase could take another molecule of acetyl-CoA and join it and make more citrate. Okay, if you look at the energetics of the TCA cycle, I think I just asked you to take it on faith that one acetyl-CoA can make 10 ATP potential. So this is part of where that number comes from. So you're getting 3 NADHs reduced. Each of those can make 2.5 ATP, so you've got 7.5 ATP. Succinate dehydrogenase, that's making an FAD2H reduced, that can make 1.5 ATP. And then the thiokinase reaction is making GTP, which is energetically equivalent to ATP. Okay, so there's where you get 10 ATP. And on Friday, I will hopefully explain where the 2.5 ATP per NADH and the 1.5 ATP from FAD come from. But if you just take it on faith for now. Okay, should we do a clicker question or two and then take a break? Okay, we're going to get 100% on this one. If we do, I'll have a question this easy on the final exam. It's my promise. You want to talk about it for a minute? Make sure you get it? No, I think it's going to be 100%. Well done. It's not going to be exactly that question, but it's going to be one that will score 100%. I'm not sure if you're allowed to get 2 answers on your thing here, but just pick your favorite one, I guess. Okay. Everybody got a chance to answer here? Okay, so citrate and isocitrate are the two tricarboxylates. Okay, I've got one more clicker question, but I think it's a good segue into the material on regulation of the TCA cycle. And given the time, why don't we take a break and resume our discussion at 9 o'clock.
Yeah, I went
Stands up in
like 7 to 10 on Wednesday morning. And then I saw Kai really briefly drop off the hobby first time yesterday morning.
But like, we'll be good. There's one out there that I'm like, I don't know. Yeah, octopus, whole, pretty common. There's like what, like that one starts with the
Alright, let's like on that.
and
Straight up.
like pretty much straight. It's like you start like really low to the ground and you like you start with the right heel. And I'm like,
Everyone else is the one.
I cannot get myself to
We're like,
One. Okay, the pink and
oh my god, we were trying to do that
yellow one,
which
too.
was like insane. Like I don't even know how, because you have to. We think that you have to go, and we can't even get just by.
so that, and then this pink, like that's the purple.
All the results
And
memorized.
that's the other one that's incredible level. And so Jamie's is he like
Hard
goes
through it.
here and then like just like glue to the right.
It's like